Welcome back to cell biology class. So we've been talking about how cells grow, how they go through the growth cycles in the cell cycle. One thing that's important is the presence of growth factors. So if you were a little amoeba, all you'd have to worry about to know if it was okay to grow was if there were nutrients around. You've got enough food, you've got enough energy, so it's safe to grow. But as a multicellular organism, any single cell can't just say, oh, there's plenty of food, I'll start growing. It all has to be controlled to be for the best outcome for the organism as a whole. So sometimes some cells aren't needed to grow, even if there are plenty of nutrients around. So cells in a multicellular organism like us are going to require not only nutrients, but also growth factors before they're able to start proliferating. Sometimes growth factors are called mitogens, just a little signal that they are related to mitosis generation. If you're a cell in a mammal that has plenty of nutrients around, but there aren't growth factors, you are going to be arrested in G1. So you'll just kind of opt out of the cell cycle until those growth factors are around. Growth and division can be triggered by adding blood serum because blood serum has different stimulatory growth factors. That means things that are going to stimulate growth. One of those would be PDGF, or platelet-derived growth factor, and another would be epidermal growth factor. Different types of growth factors are going to be needed for different types of cells to trigger them to be able to grow. Growth factors like PDGF or EGF are going to bind to a receptor on the outside of its target cell, and that receptor is going to then inside the cell activate a tyrosine kinase receptors. And this is going to trigger this whole cascade to go on through the cell. So tyrosine kinase receptors are going to activate, in particular, the RAS pathway. So these kinases add phosphate groups, so they're going to phosphorylate RAS, and RAS is going to then activate RAF. So the growth factor has been bound to the receptor, you've activated RAS. RAS is going to lead to the activation of RAF, and that's going to start a whole phosphorylation cascade of all of these other things. RAF is going to phosphorylate MEK, so you have phosphorylated MEK, and MEK phosphorylates MAP kinase, or mitogen activated protein kinase. So now you have phosphorylated MAP kinase, which is able to go into the nucleus. When MAP kinase goes into the nucleus, it's going to start phosphorylating specific genes. So Jun is going to be phosphorylated, which is going to trigger the activation of early genes. Early genes are going to include things like MYC, Jun, and Phos. And these are genes that are going to lead to transcription of delayed genes. Some of these delayed genes include E2F, that's a transcription factor we talked about earlier, cyclin, CDK. So we already talked about how all of these are involved in regulating the cell cycle, but it has to be stimulated by this growth factor first. So the growth factor triggers the tyrosine kinases, which trigger RAS, RAF, MEK gets phosphorylated, MAP kinase gets phosphorylated, goes into the nucleus, triggers Jun, turn on early genes, turn on delayed genes, E2F, cyclin, and CDK. And then you have growth through the cell cycle. Activated growth factors also can go through other pathways, not just the RAS pathway. One of them is this PI3-Kinase AKT pathway. So when the growth factor binds to the receptor, that is going to trigger PI3-Kinase to be activated. PI3-Kinase here stands for phosphatidylinositol 3 kinase, but PI3-Kinase is sufficient. PI3-Kinase is going to turn on PIP3. PIP3 is a fun little name. That stands for phosphatidylinositol 3,4,5 phosphate, which is going to lead to AKT being phosphorylated. AKT is going to signal through BAD to inhibit cell death, to inhibit apoptosis. It's also going to go through REB, activate REB, which activates TOR. Remember we talked about TOR already, which is going to increase cell growth. So this growth factor has two effects it has on the cell through activating PI3 kinase. One is preventing cell death, and one is encouraging cell growth. There's also, interestingly enough, some growth factors that inhibit growth. So not all growth factors stimulate a cell to grow. Some actually inhibit it. One example would be TGF-beta, or transforming growth factor beta. This little cursive-y looking B is a beta. So TGF-beta, depending on what cell, what receptor it's binding to, could be sometimes activating and sometimes inhibitory. The one way it can be inhibitory is if TGF-beta binding to its receptor phosphorylates SMAD. SMAD moves into the nucleus, and it will turn on genes that inhibit cell proliferation. So TGF-beta, SMAD, inhibiting proliferation. Two of these targets that will inhibit proliferation would be CDK inhibitors that block cell cycle progression, so P15 and P21. Okay, so TGF-beta binds the receptor, phosphorylates SMAD, moves to the nucleus, activates genes that inhibit proliferation, like P15 and P21. Okay, so then to put it all together, we have this whole big machine that is regulating our cell cycle. All right, we are going to sit here and make sure that we're going through G1, through the restriction point, through DNA synthesis, through G2, through the G2M transition, through mitosis. And all of these sort of regulatory machine that helps it all work. So we have this autonomous clock that's going through the same fixed cycle over and over again. And the way it's able to go through that is because, one, we are activating and degrading cyclins that are going to allow CDKs to bind to cyclins and be activated. And the clock can be adjusted when it needs by feedback from the internal and external environments, things like double-stranded DNA breaks or growth factors. And often those are going to work through kinases and phosphatases. So adding and taking off phosphate groups are going to either activate or deactivate some of our proteins and either trigger things to be degraded or trigger them to be activated. Okay, so that's the end of this one. I'll see you back for our last cell cycle video talking about apoptosis.